right, so we've had a series of great talks, and we will conclude with another great talk. We're very happy to have Kun-Hsing, who was an MD from National Taiwan University, PhD from Stanford, and is now assistant professor in our Harvard Medical School, and has won many awards for his research there. So let's welcome Kun-Hsing.
Thank you so much for the introduction, Michael. So I tend to speak a bit softly toward the end of the day, so I hope people at the back will still hear me okay. Can people hear me all right on the back? Fantastic. So I know everyone has been looking forward to my talk because this is the last one of the day. And I hope I would leave you with some impressions about some exciting applications on cutting-edge mathematical innovation related to neural network when applying to cancer diagnosis. So the title of my talk is Foundation Models for Real-Time Cancer Diagnosis. And the overarching goal of my lab is to incorporate new artificial intelligence methods for integrating three types of data. The first type is morphology. By morphology, we are using high-resolution digital scans of human tissue under the microscope. And we use AI and machine learning-based approach to analyze those human tissue at high resolution. So how does those tissue look like? Essentially, under the microscope, we would be able to identify the cells and the interactions between the different cells under the microscope. And this is how pathologists, especially within the medical practicing domain, diagnose many types of cancers. And recently, we also incorporate super resolution microscopy, which is a new technique which allows us to look inside of the cells to identify the morphology in different components within the cell body and cell nucleus. And the second type of medical data we are using is related to multi-omics. So this is a series of data that all end with the word omics, including genomics, epigenomics, which is a regulation of the genomics data, transcriptomics, which is about the expression of the gene activities, and proteomics, that is the actual proteins that have functions inside of our cells and our body. So those additional high dimensional data would help us to profile the molecular signals within the same biological sample. And here we use weakly supervised machine learning to connect these two types of data. For example, we can use morphology as the input, build a machine learning model to predict important multi-omics operations. And we can also do this in another way, where we build a model taking multi-omics, the molecular data as the input, as the predictive features to predict important morphologies we are expected to see under the microscope. And lastly, we connect both morphological data and multi-omics information to make medical applications. For example, we can build a multi-modality machine learning approach, leveraging both imaging and molecular data to make better disease diagnosis, prognosis, which is about the prediction of the patient survival and patient outcomes. And we can use the predicted treatment response and survival information to optimize treatments for individual patients. So there are a few challenges when we apply this AI and machine learning-based approach to analyze this type of data. First of all, the dimensionality of the imaging and molecular data is huge. For the high-resolution scans of the human tissue, we routinely get more than 1 billion pixels just within one sample. And for many of these data, we also have incomplete labels and labels with variable qualities. For example, if we collect the pathology reports from multiple hospitals worldwide, there's no guarantee that the levels of detail for those human annotation on the images will be the same. So there will be some batch effects, some biases related if we just naively take all the available data for routine analysis. And third, many recent research shows that there will be limited model interpretability and generalizability if we simply apply off-the-shelf machine learning models. These models are largely built as black boxes because they have a lot of parameters, and they don't easily connect with our prior knowledge about this human tissue and molecular information. And lastly, the topic about algorithmic fairness is about analyzing whether the model would inevitably discriminate against different populations because they have differential performance in different population subgroups. And that is also a specific challenge in this domain. And our solution is to develop and apply scalable machine learning approaches for multi-modality data fusion. For example, as we mentioned here, in these three different modalities of data, they all look very different, and we need scalable methods to incorporate this high-dimensional information for making predictions. And to address the incomplete label problem, we use weakly supervised machine learning. And to address the limited interpretability, we use causal machine learning and interpretable machine learning to connect our background knowledge about how this tissue should look like with the AI-based feature extraction. And lastly, we rigorously evaluate our machine learning models in diverse populations to identify and address issues related to algorithmic fairness. And for today's talk, I would only highlight two major parts in our recent investigation. The first one is a multi-modality AI foundation model for multi-omics, the molecular data, and survival outcome prediction using high-resolution medical imaging. And the second one is related to a series of applications that we can deploy in the real world to enhance cancer patients' treatment and diagnosis. And lastly, I will cover some of the challenges that we still face as a field and some potential future directions and the road ahead that we can work together in this multidisciplinary research. So I will start with the first one, the multi-modal AI foundation system. So to understand the clinical question we are facing, so let's start with the really basics. How do clinicians diagnose cancers? How do they identify different types of cancers requiring different forms of treatment? The procedure will usually involve taking a piece of the sample from our cancer patients and process the sample into thin microscopic slides, stain them with appropriate chemicals, and just observe them under the microscope. So based on the experience and the expertise of our trained clinicians, and more specifically, the specialists in pathology called pathologists, based on their evaluation, they will provide us with the diagnosis and the disease subtypes. But this process can be fairly laborious because in this approach, we have to have some experienced human being to read the microscopic slide, basically reviewing billions of pixels to arrive at their final diagnosis. So to address this issue, since a few decades ago, many computer scientists have been developing computational imaging processing pipelines to enable the extraction of objective features and to enhance diagnosis this way. But this attempt of automation turned out to be fairly difficult, largely because it's really difficult to define the exact features useful for our diagnostic purposes. And for many experienced pathologists, they are not looking for specific features, but instead the gestalt or the overall architecture in these high-resolution images. And those gestalt or the high-level view or the high-level feeling about the samples we are handling is not easily codified into computational systems. So to address this issue, here we developed a foundation model. Basically, it's a large model incorporating a tremendous amount of data in the training and the pre-training process. And we call this model the Clinical Histopathology Imaging Evaluation Foundation Model, or CHIF model in short. So to enable this process, first we collected several thousand of whole site pathology images from multiple anatomical sites, basically from multiple parts of the human body from our pathology archives. Basically, we visited several hospitals, gathered the data that they have collected in the recent 10 years, and we digitized them into this digital format. And as we have mentioned, because many of these tissue under the microscope, they look huge. In the real world, the size is probably only around 1 inch by 3 inch. But under high-resolution microscope, we can generate billions of pixels from there. So it's not easily applicable if we just dump all this information into an off-the-shelf machine learning model like those inspired by ImageNet. So we have to do something more creative. And our approach here is starting from the whole slide image, this high resolution view with billions of pixels, we first pile them into individual image patches, and then we feed them into some image encoders to extract features from those images. And we also leverage another source of information in our multi-modality analysis. That will be the text descriptions of the same samples. So from our text descriptions, we can identify a few useful information from our pathology reports written by pathology experts. But one caveat is that not all samples are accompanied by very detailed description of that particular human tissue. So our approach is we just take some simple description that we can reliably identify from all sources. And those are largely related to the description of where this particular sample is coming from. That is, which human organ this particular tissue came from. And we put the extracted text into a text encoder, and we use the clip-based embedding to extract the embeddings from there. The clip model is a multi-modality model developed by OpenAI, and the goal is to align the text-based descriptions with the image-based features. So once we have extracted the text-based descriptions, we aggregate them with the image-based features for further analysis. And for this paper, we connected these features with 3 different forms of important clinical evaluation tasks. The first one is related to diagnosis, that is, what kind of disease this particular patient suffers from. And the second group is about molecular prediction, essentially predicting what type of genetic variations our patient have, and we can use those pieces of information to recommend the most suitable treatment for individual patients. And the last one is on survival prediction, predicting the treatment response and how long would our patient survive under the standard treatment. To train this foundation model, we have incorporated more than 60,000 whole slide images. And after extracting the image tiles, we have more than 900 million image patches to train this pathology foundation model. And this data is collected from more than 19 parts of anatomical site, is basically different parts of the human body. And this large-scale analysis is not accomplishable without our team. We have 5 talented students and postdocs in my group working on this project. They are Kun-Hsing Zhao, Xirue Wang, Eliana Marustica, Chris Jackson, and Sen Yang. And how does this model look like, and what's inside of this giant model? So this is something slightly more technical here, and I'm sure some folks in this audience will be interested in this. So essentially, starting from our image features, we first extract the image patches or the image tiles, as we mentioned, and we feed them into a neural network. And more specifically, this is a residual neural network-based design, as the previous speaker has covered. So here we have multiple layers of neural network to extract the intermediate signals, pass them through the rectified linear unit as usual, and then we have different pipelines of the image processing to use different activation functions, and then we aggregate them all together. And then at the end, we also extracted the attention labels for evaluating these slides, and then we connect them with the original input information into another tensor multiplication. So this information highway right here is inspired by residual neural network. It's essentially a skip connection to allow some of the image features we have identified using simple image encoders that is based on a previous published pipeline called C-Transpath. And we aggregate them with our additional layers of neural networks extracted features for further analysis. And at the end, we train a model based on multi-instance learning. This is largely because within the same whole slide image, here we only request the pathologist to identify the final diagnosis for the entire set of samples without specifically labeling which part has the tumor tissue and which part does not have the tumor tissue, and just being identified as adjacent normal samples from the same patient. So because of that, we are entering a domain where our positive samples would have some regions with cancers, but not every single cell in that particular sample would belong to the cancerous category. But in contrast, if a particular sample is labeled as normal, then every single cell there should be completely normal. So to solve this problem without strong supervision, where we would have to ask a bunch of experts to relabel every single cell for us, here we just look at the sample label annotation, whether this entire sample was labeled as cancerous sample, meaning that there would be some abnormal cells there, or this entire sample was labeled as completely normal. So with that, we use a pretty standard approach of multi-instance learning to compute the loss function there. So suppose we have infinite sample size, and we also have optimized models. This pipeline should be sufficient in training a performing model. But in reality, there are a few details related to the noise of the data, and also the amount of information may not be sufficient in training a good performing model. So we further utilize two additional modules, most related to the attention refinement. The first module is related to instance level classification. So essentially, we train another model to simply identify the regions require high attention, meaning that those regions may be interesting and informative in defining cancers. And in contrast, there would be some low attention tiles, the regions that are not so useful in helping us to make the final diagnosis. For example, those could be just the blank backgrounds or the tissue that doesn't contain any diagnostic information. So here we train a simple binary classifier to help us identify the regions requiring additional attention versus the regions that we can safely skip. And we further incorporate information from this instance level module back into our attention learning system. And the last module here is the whole slide image level module. So here we use contrastive learning to compare our task at hand, the input slide, the input sample that we are trying to classify as the anchor bag, and we compare it with our existing features extracted from the previous batch of the samples. So essentially here we define three types of subset and store them in our memory bank. The positive set will be the high attention regions for the cases labeled as positive in the same data set. And the negative memory bank will be the regions that have received high attention among the negative cases. That is a hard negative memory bank. Those will be the high attention regions of the samples labeled as negative. And then in the middle, the negative memory bank will be the regions coming from the negative classes, but they receive low attention. So this will be an approach to further identify the defining patterns of a positive case in a positive memory bank and the defining factors in a negative case that we put in the hard negative memory bank. And the negative memory bank, that is the third memory bank, will just store the images receiving low attention in the negative samples to help us identify which regions we can safely skip, because we know for sure those regions doesn't have a cancer cell at all, and also they don't require additional attention in our further analysis. And at the end of the day, our total loss function that we use to train our entire model would be just the summation of the loss from our multi-instance learning approach, that is this end-to-end pipeline, plus lambda times the instance level loss, and plus the lambda 2 times the loss of this whole slide level module. And lambda 1 and lambda 2 are just additional hyperparameters that we can optimize and fine-tune. So these are some technical details about this foundation model. And how does our result look like? Using this foundation model, we show that we are able to outperform all existing methods in cancer cell detection tasks. So for this task, our goal is to identify whether this sample contains a cancer cell or not. And this is very important because if a patient has a tissue coming, even just one single cancer cell, we would require some attention, require some additional evaluation, if not treatment. And in this result, we are showing the standard ROC curve, the receiver operating characteristic curve, where the x-axis is 1 minus specificity, and the x-axis is 1 minus specificity, and y-axis is the sensitivity. So the closer to the upper left corner means the model has better performance. And we show that our method, shown in red, we have near perfect performance in detecting cancer cells within many types of cancers, including colorectal cancer, esophageal cancer, stomach cancer, and many others listed in this figure. And comparing with many prior published approaches, we have an improvement of around 10 to 15% in the area under the curve in the cancer detection task. And interestingly, yes.
Question about the previous slide.
Yeah.
So you mentioned like you did have like annotations for those tiles to make sure we know which one responds to cancerous region, which one doesn't, right? When you show the next graph on the comparison between the performance, do the other models like use the same annotated like tiles and frames to train, or was it like the original picture that was just annotated as like cancerous or not cancerous? So how was it compared?
Yeah, that's a great point. So the question is about, in this comparison, do we use the same set of annotation? And the answer is yes. So here we only use the whole slide label annotation, basically the sample label annotation for all methods, including our method. And how does it work? So first of all, we extract the tiles, but we don't have the annotation for each tile. That would be really labor intensive. And our approach is first allow all tiles to inherit the labels from the sample label. So if a sample is labeled as cancerous, here we just temporarily put all the tiles from those samples as cancerous. And for the sample that was labeled as completely normal, we have all the tiles labeled as normal. And this would have a problem in the multi-instance learning that we have just mentioned. And our solution, largely based on this whole site level module, use constructive learning to differentiate. Within the positive cases, there would be only a really small subset of regions containing cancer cells, and we use that. We store that in a positive memory bank by first identifying which regions receive higher attention, and we put them in this memory bank, basically a storage repository of typical cases labeled as positive. And we do the same thing for negative in the hard negative memory bank, and for negative we further have another storage grid for so-called negative memory bank, meaning the low attention regions of the completely normal samples. So this is our approach to address this imperfect labeling when we inherit the sample label to each individual tile. And we use the same approach for all comparison methods.
And just to check on the next slide, when you compare all the models, you're rerunning all the other models on your data set.
Exactly. Yeah. So here we show the result from 15 different data sets, and we just run the same model, run the same data set, the same training partition, same validation, and same test partition to make our comparison. Those are very good points. So here we show that we can outperform many existing models in the cancer detection task. And one interesting finding is related to this question. How does our model learn, and can we use this model to help us make instance label or the region label annotation? And the short answer is yes, and this was quite magical to us when we first saw this. So now we have a model that would recognize whether a particular sample contains cancer cell or not. And now we can use this model to help us annotate each individual pixel in our original image. So our approach here is we just input separate patches and show them as independent samples to our model, ask it to produce the probability that this region contains cancer cell. So we use that to annotate the entire sample without the help of any human input. And somewhat surprisingly, when we compare the annotation from expert pathologists on the same sample, those are generated separately. And after the completion of our model training process, and we ask a few pathologists to label selected samples, use this green outline to identify the regions truly occupied by cancer cells. And we show a substantial concordance between our model labeled heat map in red, those high probability cancerous regions with the ground truth annotations independently made by pathologists. Here now we can apply this sample label classifier as a region label annotator to annotate each individual region or each individual pixels in our original samples. So this is about cancer cell identification, and we further challenge our model to do something even more challenging and also very clinically relevant. So one of the current challenges about identifying the origins of cancers when we first diagnose the patient, the cancer is already everywhere, spreading to the brain, to the lung, to different parts of the body. And when this happens, it's really hard to identify where the origin of the cancer is coming from, whether it's lung cancer spread to the brain or brain cancer spread back to the lung. And because cancer treatment depend a lot on their origin, different sources of the cancer, they still have different behavior. So identify the origin of cancer is very important, especially for the patient. When we first encounter them, they are already at the end stage. So to solve this clinical challenge, we apply our chief model to see if we can identify the sources of their cancer origin. And the short answer here is yes, we can directly use the features extracted by chief and then fine-tune a model to do a K-way classification. And in our analysis, here we have 18 different types of cancers with solid information on where that origin truly comes from. And we use this to train and validate our model. We show that our approach can achieve, again, a near perfect performance of an area under the curve of 0.996 in classifying the origins of cancer in this 18-way classification. And we show that for most of the cases, we've been able to correctly identify their true origin. And among a few cases where we cannot, we also see an interesting correlation between our erroneous prediction versus the actual origin. For example, in the cases of truly arising from the colorectal region of the cancers, 97% of cases we identify correctly, the source is truly colorectal. But among the last 3%, our predictions are related to other parts of the gastrointestinal tract. So for example, 1% was erroneously classified as esophageal cancer, the other 1% coming from stomach cancer. So showing that we are seeing some similarities across the cancer types, and we use that as a source of information for predicting the origins of cancers. So this is about the important diagnostic task in the clinical settings. And the next question is, can we further identify additional hidden information on our histopathology images? For example, can we read out some hidden genomic information by just looking at the image? So as an analogy, here we have a picture. Simply consists of just some random log in the forest. And the question to all of you is, can we identify some hidden signals, some hidden animals inside of this particular image? So we just look at it closely. Can we see the hidden animal here? It seems to be a pretty difficult task because it seems at the first glance like everything is just the random log. But what if we look closely, and probably we also need to squint a little, we would identify the hidden cat inside of these random piles of wood. So the analogy here is when we look at pathology samples in our clinical practice, there are tons of information that we know how to utilize to, for example, identify cells and identify things that we already know related to cancer classification. But what if there are additional hidden information inside of this tissue that is just not well documented in the literature, such that we don't know how to predict them? For example, can we just look at the image and then predict what type of genomic variations our patient have? So this is way beyond the human capability, and so neither me or my colleagues will be able to predict their genomic variation by just looking at the pathology samples. So we put this challenging task to our chief model, our AI model, and see how our model says. And surprisingly, among the prevalent mutations, so those are the mutations with greater than 5% of prevalence rate in a cancerous population, we show that we can reliably predict many types of mutations, genetic mutations, by just looking at the figure, the graph under the microscope. So for example, in predicting the GTF2I gene among thymoma, we have a 91% area under the curve in such prediction. And more clinically interestingly, many mutations are directly related to FDA-approved targeted therapy drugs. And here we show that for many of these mutations, we also have pretty good performance in predicting them. For example, when looking at a type of lymphoma tissue, we can predict the mutation of EZH2, which is a gene related to the targeted therapy approved for this cancer, with 95% of area under the curve. So this shows that our AI-based approach can exceed the human capability and further identify hidden signals related to genomic variations.
Using this by scanning for a much longer list of potential mutations or select the candidates in some other way.
Yeah. So the question is about how do we select these mutations? So for the figure on the left, we just pick all mutations with high prevalence, and by high prevalence we mean more than 5% of prevalence within each individual cancer types. So it's a systematic analysis, and here we show all the results here. So if we look at it closely, for certain genetic variations, we are not doing so well. For example, at the very bottom, for this particular type of genetic variations, also with biological interest, among uterine cancers, we only got an area under the curve of only 0.54. So showing that certain genes are easier to predict by looking at the image, but others are much harder. And perhaps it's an issue of whether we actually have the signals from the tissue, or it could be that our current model is still not robust enough in identifying those additional hidden signals. So that is a very good question. We see a wide range of performance across genes when we do a systematic analysis. But interestingly, for many types of mutations related to treatment, perhaps they are more directly related to the replication and the growth of tumor cells. So on average, we seem to have slightly higher performance. So this is about the genetic variation prediction, where we connect our imaging patterns with the molecular domain, which will not be visible to the human eyes under standard light microscopy. And next, we further extended our approach to predict the molecular profiles response related to the response to novel treatment, including immunotherapy or immune checkpoint blockade, to be specific. So there are many biomarkers and many molecular profiles related to the response of such novel treatment, which won a Nobel Prize in 2018 in physiology and medicine. And we show that one of the well-established biomarker, microsatellite instability, or MSI, we have fairly good prediction among colorectal cancer patients with area under the curve of more than 0.86 across different populations. And one interesting bit about our interpretable machine learning approach is that we can further connect them with our prior knowledge on what is abundant in our tumor cell and how do they correspond to the predictions of the treatment response. So here we show that in predicting the status of microsatellite instability, this biomarker for treatment, we show that lymphocyte percentage is a very big predictor for this particular treatment response, with a statistical significant difference between those that would be responsive to the treatment versus those who cannot. And we further see some differential abundance in the stromal cell. Those are the supporting cells in the same tissues as well. So this shows that we can connect our machine-derived features back to our biological background knowledge about what defines the tissue and what we can utilize to predict patient's treatment response. And lastly, about this paper, we show that our approach can also predict cancer patient survival outcomes. So to predict individual patient survival outcome is a very difficult task because there are so many different factors that we need to consider, including the general health status, the comorbidities, like other diseases the same patient may have. So in order to simplify this problem and to make it statistically valid, here we do an approach where we stratify our patients into two groups, one with predicted better survival, the other with predicted poor survival. And in these survival curves, on the x-axis, we show the survival time after the initial diagnosis. So time D will be the time of initial diagnosis aligned for all patients. And we start to follow up with the patients after they receive the standard treatment. On the y-axis, we plot the survival probability. That is, how many percent of patients within that group have survived for a certain time period. So as time goes on, the survival probability would be from 100% at day zero, and we will start to observe some mortality. So the survival probability will always drop, never go up in these survival curves. And as we can see here, our approach successfully differentiated two types of patients, one shown in blue, the other shown in red. These two groups of patients have differential survival probability and differential mortality risk. And we conducted a log rank test, shows that in all the cancer types and all the sample set that we have reliable survival information, we can differentiate the blue group from the red group, the differential survival group, with statistically significant difference. And we further compared our approach with other methods, and we compare in terms of the C index, the concordance index, which is about how concordant we are in our prediction versus the actual observation that we see in our patient. For example, when comparing two patients, if our model says that patient A would have a longer survival probability than patient B, then when we look at the actual data, does that still hold true in our observation? So using that, we can compute the standard concordance index. And here we show that our approach labeled in this orange color, we have better performance than many other approaches throughout different test sets and different cancer types. And we further identify the regions related to those occupied by our high attention regions. And we show that those are largely related to dense clusters of cancer cells, which also align with our prior biological knowledge.
Another quick question about the previous slide. Yeah. So data that you used were based on patients that did not receive any sort of like treatment, and you analyzed and trained on their survival rate. So there is no intervention for those patients.
Yeah, that's a good question. So for all our observational analysis, we didn't do any additional intervention. So all our patients would receive the standard treatment. So we collect this patient retrospectively when everyone has been diagnosed and treated under the standard clinical routine. And after all those data has been collected, starting from around like 1.5 years ago, we started to initiate this analysis by digging into our data set. So all these patients, after the original diagnosis from time zero, they would start to receive the standard treatment. For example, among the first one, breast cancer, in the lower stage breast cancer, stage 1 and stage 2, surgery would be the first line treatment. And all of these patients would receive some form of standard treatment. And you also hinted at a very good point related to this treatment question. It's about the treatment heterogeneity. Because sometimes our patient, especially the higher stage patient, they don't always receive the same set of treatment. Some may be enrolled in clinical trial and receive some experimental form of drugs, and others may be receiving some combinations of chemotherapy, radiotherapy, and others. So among the higher stage cancer patient is a much more heterogeneous patient group. And in the results shown here, it's only stage 1 and stage 2 combined. In our supplemental figure for the same paper, we showed the stage 3 as a group and stage 4 as a group. And there we do see a much more heterogeneity. But the bottom line is our model are still be able to predict the survival groups with statistically significant difference in the blue and red groups in most sub-analyses.
So that will also predict the stage.
That's another good point. In our paper, we didn't specifically predict the stage because the stage will require a holistic analysis on the extent of the tumor spread. For example, if the same lung cancer tissue stay in the lung, it's usually stage 1 or 2. But if it has spread into any other parts of the body, to the liver, to the brain, it's by definition stage 4.
That's identified. That's information or something to.
Yes. Yeah, that's right. So we have all those information ready. And in the current standard analysis, doctors would have to resort to other information. For example, whole body CT scan or whole body MRI to identify the distant metastasis in order to define the final stage.
My question was that on the other side of what you pointed out that there might be hidden clues.
Yes.
That you put in the slide, perhaps those hidden clues, perhaps they can help predict the stage, perhaps they cannot help predict the stages.
Yeah.
That's a question for this study.
Yeah, that's right. So in this paper, we didn't address that question, and in one of our previous paper, we used the renal cancer data, and we see that we can provide some hint to the stage with the area under the curve of very modest, around like 0.7 to 0.75. So it's not clinically usable yet, but that's also provides some clue showing that our high resolution image contains some additional information which would be incorporated. That's a very good point and not addressed in this particular paper. Great. So as we shown here, so our neural network-based approach, this shift model, has predicted survival outcomes and also automatically identified the regions related to the patient survival. And after we published this paper, it's just shown up online the past Wednesday. We received a few public media press. So many of these descriptions does not have any clinical jargon, and feel free to read some of these summaries of our most recent paper. So after publishing this paper in the flagship journal of Nature, we've received a few ongoing requests on how do we implement this to the real world to assist with the real world clinical diagnosis. And in the interest of time, I will probably just cover the first one about real-time brain cancer evaluation using a related model that we published previously. So in this approach, we are addressing a particularly interesting challenge in the clinical diagnosis about brain cancer. As many of you know, brain cancer remains a huge healthcare problem worldwide and contribute to more than 200,000 deaths per year worldwide. And in the diagnosis part, usually it's suspected by signs and symptoms and also MRI of the brain. And once there's a suspicion that we are handling a brain cancer case, we usually have a surgery to extract samples from our patient for further analysis. And in this process, we have neuropathologists to stand by to evaluate those tumor samples when the surgery is ongoing. Because suppose, surprisingly, that suspected tumor region does not contain cancer. For example, it could be just benign tumor, or it's not a tumor at all. It's just that the clinical behavior mimic those of a cancer. Then we probably wouldn't proceed with the surgery, and instead we would close the surgery and then resort to the most suitable treatment depending on the final diagnosis from the pathologist. And suppose the sample we obtain is truly a tumor sample, then another important thing pathologists need to determine on the fly would be what's the extent of the tumor, what's the boundary of the tumor, because we want to cut off as much tumor as possible that would be beneficial to the patient prognosis. So this approach will be doing surgery. When the surgery is ongoing, we send a piece of tumor sample to the Department of Pathology, and the pathologist would not have the time to process the sample using the standard procedure of fixing them in formalins and pass to the standard microtome for evaluation. And instead, they would just put the sample into liquid nitrogen, make it stiff enough to make them into microscopic slides, and then observe those lower quality samples on the fly. And they need to report back to the surgical team, usually within 10 to 15 minutes, because the surgery is still ongoing and they're waiting for the diagnostic results. So this process, as you can see, is pretty costly because we always need a neuropathologist team to stand by. And when we are doing this 10 to 15 minutes of evaluation, the surgical team essentially have nothing to do. So we always say, how do they do that? They probably would have to just gas it around because they are forced to, or if allowed, they would love to have a bottle of beer, but it's not allowed to have beer in the operation room. So there's really no good solution to this problem currently. And because the pathology team is under time pressure, they need to provide the right diagnosis within 10 to 15 minutes. So it's also contribute to higher human error rate, especially they are evaluating the imperfect sample from so-called frozen section, the liquid nitrogen-based section tissue. And this both contribute to patient burnout on both ends. So our method is we leverage a related form of AI-based approach for cryo-section-based pathology analysis. So in this procedure, we again start with a whole site image, and we identify the tiles, the tissue patches likely contain diagnostic signals. We partition the data set and also incorporate independent validation set from different hospitals. And we train a hierarchical vision transformer to train and validate our model. And lastly, we aggregate all tiles from our samples to make patient-level predictions. Because at the end of the day, clinicians only need one decision, whether this patient is either have cancer or not, and whether this will be a high-grade tumor, require more aggressive surgery or not. So eventually, we provide the aggregated prediction from different regions, considering different regions and their tissue context. So here we show that this different forms of AI model, also empowered by neural network, can identify the cancerous cells from our tissue samples with an accuracy of 0.94, which is on par with expert neuropathologists in this particularly challenging task. In addition, we successfully differentiated the histopathology grade using the same approach, showing that we would be able to identify the cases with more aggressive form of tumor requiring more aggressive surgical resection, and the tolerance for residual tumor for those cases will be lower. So this tool can be used to guide real-time surgical decision in the operation room. And we are currently piloting implementing this model in our affiliated hospitals and our collaborative hospitals to further validate the real-world impact of implementing such AI models in the real-world settings. So in the interest of time, I think I would skip through some of the other examples and just jump to the conclusion. So in conclusion, here we show that we have a multi-modal foundation model that we have recently published to identify patient diagnosis, molecular subtype, and prognosis using just the high-resolution digital pathology images. And we further show that our AI-based approach can empower clinical decision-making in real time with a particular use case of brain cancer real-time diagnosis during surgery. And because this field still encounter a few additional challenges, as we first mentioned in the second slide, multidisciplinary research is still required to make the most of the useful information from our high-resolution microscopic slides and additional molecular data to make the most of this information for clinical practice. And lastly, many people ask us, what will be the future of AI in medicine? And we don't really have a good answer because it's really hard to predict what would be the next big thing in the field of AI, what will be the next big invention here. So no one really knows for sure what the future would look like. However, there's one thing we know with certainty, that is, the best way to predict the future is to create it. And we would like to invite all of you to join our multidisciplinary journey to create a better future of AI in medicine. Thank you.